think everybody who is going to be here right now is here. So we'll go ahead and get started. So today I would like to introduce you guys to Matthew Hilliard of Danimer Scientific. So Matthew grew up in Pelham, Georgia, or Pelham. Am I saying it's Pelham, right?
Pelham, yes.
Yeah. Where he attended Pelham City Schools and graduated from Pelham High School in 2009. Following graduation, he pursued a bachelor's degree in chemistry at Georgia College and State University in Milledgeville, Georgia. While in Milledgeville, he fell in love with the campus and being in college, so he decided to stick around for 5 years and graduate as a triple major with a bachelor's degree in chemistry, mathematics, and physics. He wasn't quite sure what he wanted to do after graduation, so he decided to continue schooling, pursuing a PhD in chemical engineering at Auburn University. He completed his PhD in May 2019 and started a biotechnology startup company that is developing technology to better treat wastewater and mitigate carbon dioxide and methane emissions from farms and wastewater treatment facilities. In January 2021, he joined Danimer Scientific in Bainbridge, Georgia, where he currently works in the microbiology lab.
All right, perfect. Thank you, Megan, for the introduction, and thank you, Megan and Kristen, for the opportunity to come and talk to you and the BioVet Institute for supporting this. And then thank you to all the teachers for attending. I really appreciate the investment that you're making, hopefully in your students' lives through participating in training programs such as this one. So I'll go ahead and start. Hopefully, I need to share my screen first, don't I? Let's see, share screen. I'll just do screen. F5, that didn't work, so we'll go down here. All right, perfect. So like Megan said, my name is Matthew Hilliard. I am a microbiologist here at Danimer Scientific. I actually joined Danimer not too long ago, a couple months ago, January of this year. So I'm relatively new to the company, but not new to the area and certainly not new to the field. So I'm happy to be here and again, talk to you. Again, to kind of go back over what Megan said in the introduction, I am from Pelham, Georgia. I grew up there, graduated high school from Pelham High School, went off to Milledgeville, and like I said, enjoyed my time there. Ended up doing the 3 degrees. Wasn't sure what I wanted to do afterwards, but I was for sure it wasn't biology. And so now I'm in the biology field and I'm very much enjoying it. So did my PhD in chemical engineering, but really was a focus in biochemical engineering, really understanding what's happening at the metabolic level of different microorganisms and how we can translate that into industrially relevant outcomes. I do have a startup company that I'm a co-founder of that looks at using a consortium of bacteria and microalgae for treating wastewater. It's still in the very early stages, but that's separate from what we do here at Danimer, which is, again, a bioplastics. And I'll talk a little bit more about who Danimer is. And so really, our mission statement is out of a concern for the environment. We're providing sustainable biopolymer solutions to plastic pollution. And that's being accomplished through innovative technologies. And I really think it's kind of funny that our motto is the future of plastics is here, because you really, maybe from what you've been teaching in your schools, you don't really hope there's too much future for plastics, because there's all this plastic pollution that we see, especially when we talk about all the single-use plastic straws, water bottles, and things like that, and they're polluting not only our local environment, but the oceans and having detrimental impacts to the environment. So really, we don't really see a future for plastics, but we do see a future for bioplastics. So quickly, who Danimer is. We were started in 2004. We're located here. We're headquartered here in Bainbridge, Georgia. I think we originally were Meridian Bioplastics, and we've since kind of switched over to Danimer Scientific. We are the world's leading producer of polyhydroxyalkanoate, also known as PHA. And the reason this is the future of plastics and bioplastics is because even though it has similar properties to traditional petroleum plastic, it is 100% biodegradable, not only in soil, but also marine biodegradable. So if these bioplastic products end up in the ocean, they will naturally biodegrade over a period of time. So PHA is really a polymer of a bunch of different monomers and really those monomers can have different shapes and sizes, kind of what I've shown here, where you have this traditional backbone, but our side chains represent these different monomers, and really the length of the side chain has a significant impact on the property of our polymers. So we primarily focus on what are called mid-chain length, which starts with, I lost my mouse point here, this hexanoate and goes up to the dodecanoate. And again, the longer these side chains are, the different properties that are associated with the plastics we can produce from this polymer. So it really is a bioplastic. It's twofold bioplastic because it has a renewable bio source, and then it's also biodegradable. So it comes from the environment and then it returns naturally to the environment. And so the way that works is we start with a carbon feedstock. Primarily, we use canola oil. And so we take the seeds from, we don't take the seeds, we actually buy the oil, but the way that it would work is we would get the seeds from farmers. They would get pressed into the oil. And we use those oils with our proprietary microorganism that's able to ferment those oils and convert that oil into our PHA, our polyhydroxyalkanoate. Then we have to separate the PHA from the cells, and we extract that PHA. We do some further downstream processing to refine that and make it into the desirable plastic resin that is going to be used in a variety of different applications. So on the right-hand side, you kind of see how it's a circular economy where we start with our plant crop. We go through our process of producing the PHA. The PHA, when you're done with your water bottle, you're done with your straw, it ends up maybe in the environment in some form or fashion. Natural existing bacteria are going to break down that PHA and convert it back into bacteria, which can again go and ends up feeding our soil, feeding our crops. And so it really is a truly circular economy. So on the left-hand side here, this image really shows you our starting materials, this canola seed. We go to our refined canola oil. This is our crude PHA product. It's a real powdery substance. And then we extrude that into our plastic resin. And this is what we ship to our customers and who are doing a variety of different applications. What all can PHA replace? I know a lot of times we use the general term plastics. However, there's multiple types of plastics, and those are all different polymers of this petroleum-based plastic. So there's polystyrene, PVC, polypropylene, and that list goes on. And PHA, as shown here in this figure, can replace all of these to a certain degree. And again, it's not one PHA product. It's the different comonomers of the PHA that allows us to replicate maybe the polystyrene characteristics or a polypropylene or something like that. And so who's interested in our products? Well, these are all, so there's several companies that we have contracts with that we're working with. Nestle being one of the largest, they're interested in using our PHA for biodegradable water bottles, which is something obviously of very significant importance because water bottles, again, in those images, make up a lot of that plastic pollution that we see, whether it's at the park or it's in a lake or in the ocean. More recently, I think either yesterday or Monday, we announced that we are partnered with Mars Wrigley for developing a biodegradable packaging for some of their products. I think the Skittles are shown here in this bottom right image. Additionally, WinCup produces what they call the Fade straw. These are biodegradable straws that are now being implemented in Dunkin' Donuts, a variety of other chain restaurants, and I unfortunately can't remember all of them now, but Dunkin' Donuts being one of the larger ones. PepsiCo, we also work with PepsiCo. You can actually buy straws made from our PHA at Walmart. This is the box, what the container looks like, and we've got some of these in our office here. Bacardi recently also is interested in developing a packaging for their products. And then the big question mark is, I mean, obviously plastics are ubiquitous in the world, and so there's endless opportunity for us to implement our PHA, our biodegradable plastic, and again, mitigate these environmental impacts that petroleum-based plastic is causing. So a little bit more, we've recently brought online the world's first commercial PHA production plant. This is in Winchester, Kentucky, where we have several thousand liter fermentation tanks that are producing tons of PHA per month. And so this is our facility in Winchester. However, I work here at the Research and Development Center in Bainbridge. We have a 20-acre campus, 200,000 square feet of manufacturing space. So our R&D houses a microbiology lab, an analytical lab. We have bench scale fermentation, pilot scale fermentation, pilot scale downstream processing, the commercial scale is also housed here. And then we also have a product development lab where they focus on developing different resins, films that can be used and that our customers can use from our PHA. So these are all of the labs that are housed here in Bainbridge. These also have these are career opportunities for some of the graduates or some of the students that you're teaching now. And I'll talk a little bit more about those opportunities later, but just wanted to show you what all is happening here locally in Bainbridge. I'm going to focus primarily on the microbiology lab because this is the lab that I work in, and I think this is most relevant to the training that y'all are doing now. So to kind of cover some of the responsibilities of our lab, we do the strain cultivation and seed stock preparation. So we're continuously developing new strains of our proprietary bacteria for different purposes, whether it produces a different length side chain, again, to kind of cater those bioplastics to the different applications. Or we also are looking at using different carbon feedstocks, and these different strains, we have to test those here in our lab, grow them up, and see how they perform compared to our other strains. More importantly, we do the seed stock preparation, which is we actually grow up the strains that we use at our commercial facility here in our lab. This is one of the main reasons that aseptic technique is so important in our lab, because these are the actual bacteria that are going to go into our fermenters up in Kentucky, and we're talking 50,000 to 100,000 liter fermentation tanks. And so if you have contamination in there, you have to shut down that large of a reactor. You're talking about a lot of lost product and a lot of lost time. So aseptic technique is extremely important. It's something that is obviously you can train the students in your classes to focus on. Other responsibilities, we do strain screening. Again, going back to these different strains have different properties, and we can screen those for different applications based on what our customer needs are. Feedstock screening, as I mentioned, our primary carbon feedstock is canola oil. However, we are looking at a variety of different carbon feedstocks ranging from sugars such as lignocellulosic biomass to corn oil and a variety of different oils. And we're trying to, again, focus on the circular economy, this sustainable economy, where we're trying to avoid using food sources as our carbon feedstock so that we, again, can maintain sustainability. Another significant job for our lab is to identify and test new materials to be used in our process. So again, I should probably have introduced this a little bit earlier. We have a two-step process, essentially what we call upstream processing, which is where we actually cultivate the bacteria in our fermenters. Inside of those fermenters is where the bacteria are actually going to produce the PHA. They store this PHA intracellularly as a extra carbon feed source, so similar to how we would store fat in our bodies, in the sense that if we ever were to, not have any food to take, we would source. They do the same thing with the PHA, to store in that PHA, getting out. So to do that, we use a variety of different enzymes working properly, that they're sending us the right stuff. And two, see if we can, oops, sorry, I have a chat here.
I think you're back now.
Okay, sorry. Yeah, it said something about connection unstable. So I'll go back just a little bit and repeat. So again, our lab's responsible for identifying and testing new materials. These are materials such as enzymes that we use to break open those cells, to remove all that cellular debris so that we can extract just our PHA product. We also use different detergents. These are surfactants that have other properties that help us to, again, clean up that, the product. We also work on protocol development. This is assisting with the different steps of either the upstream or downstream processing. We do this at the bench scale using, flask, and then we assist with scaling that up from the flask to our pilot scale, and then from pilot scale to commercial scale. Another important job is identifying and mitigating any contaminants. So again, we're growing bacteria. We're using a carbon-rich feedstock. So there's other bacteria, there's other yeast, there's other microorganisms that are capable of growing in those similar conditions. So what we have to do is make sure that we eliminate potential contaminants. And again, that goes back to our aseptic technique. In the case that there is a contaminant, we have to do, we look at them under the microscope. We use gram staining to see if we can identify what the contaminant is, the source of the contaminant, and how we can eliminate the potential, any additional contamination from these sources. So that's kind of the responsibilities of the microbiology lab. In order for us to accomplish those goals, we use a variety of different techniques almost on a daily basis. I'll start with lab safety. Is always important. It's a main focus for our lab. Fortunately, we're not working with any pathogenic microorganisms or anything that's really too dangerous, but we still require proper PPE, personal protective equipment, lab safety coats, lab goggles, closed-toe shoes, and things like that. Very, very important is the aseptic technique, where we do flame sterilization. We use the chemical sterilization, autoclaving, all of that, and then we also use laminar flow hoods or biosafety level 2 hoods to prevent any contaminants from getting into our samples and from those samples contaminating us. We do PCR, polymerase chain reaction. Many of you may be familiar with that with the current COVID-19 situation. So this is something that we do in our lab pretty frequently. Gel electrophoresis, which I believe y'all will be discussing as part of this training. And I'll talk a little bit more about how we do gel electrophoresis later in the presentation. And then I believe, if I'm not mistaken, y'all covered pipetting last week. And pipetting is obviously very important in our lab and done very frequently. As I mentioned, gram staining and microscopy, looking at the different bacteria and microorganisms under the microscope, make sure, and looking at our product under the microscope as well. Spectrophotometry, we use UV-vis spectroscopy and some other spectroscopic techniques, including fluorescence, for either testing our enzymes or testing our product or testing our bacteria. Oops, I think my screen sharing was paused. Oh, resume share, sorry. Click back here. Okay, got ahead of myself. So again, what I wanted to do was kind of go over 2 of the gel electrophoresis techniques that we utilize in our lab. One of those is sodium dodecyl sulfate polyacrylamide gel electrophoresis. Most often you'll see the acronym SDS-PAGE. So I took this, shame on me, I didn't put my source here, but I took this off the internet to really kind of just give you a general overview of how gel electrophoresis works, or for how the SDS page works. So you have these different proteins. Again, if you think about our process where we're trying to extract the PHA from within the cell, we break that cell open, and all of that cellular debris, proteins, nucleic acids are all in there. And so we need to eliminate those. And in order for us to monitor how well we're accomplishing that goal, we use SDS page. So SDS uses sodium dodecyl sulfate to help linearize these proteins. And then it also uses beta mercapta ethanol, which breaks up the disulfide bond. So you have these proteins that are these, amino acid chains that are all folded up. The SDS and the beta mercapta ethanol open that protein up, linearize it, and then we apply a electric field or a potential across our plates or our polyacromide gels. And what this does is it kind of pushes those different proteins with the negative charge down towards the cathode. So that's kind of what's shown here on the left-hand side. And then what we really want to do is separate these different proteins based on their size. And the way that's accomplished is through the polyacromide gel. It's like this gel matrix where the smaller proteins are able to navigate that matrix a lot faster than the large, high molecular weight proteins. And that allows us to separate those based on their molecular size. And so that's kind of what's shown here on the right-hand side. So to kind of show you what that looks like in action, these are some samples that I've taken recently. We're here on the left-hand side. This is sort of during our initial downstream processing. You can see we have a lot of these bands. These different bands represent different protein or different molecular weight proteins, where one band could represent one protein or could represent multiple proteins with similar molecular weights. And the way we identify what those molecular weights are is we include what we call a ladder, which is a solution of proteins of known molecular weight. And they break down based on their size, where this is, again, the highest molecular weight, and this is the lowest molecular weight. And then each of those bands represents a specific weight. And we can relate our bands to where it falls on that ladder, and that kind of gives us an idea of what molecular weight our protein is. On the left-hand side, you can see we have a lot of bands. This is kind of the initial part of the cleanup process. On the right-hand side is more our final product, where you can see the number of bands has decreased, and we're really left with these 3 residual bands. And then our final product, we have these 2 bands, and these are 37 kilodaltons and 25 kilodaltons. And what we've actually seen in our analysis and our processing is that these 2 bands are pretty persistent, which means there's a protein in our product that is a little more difficult to get rid of. And so what we're doing now is focusing on identifying, again, which enzymes or which surfactants or detergents we can use to help us eliminate those and really have a much cleaner product at the end. Additionally, we use what we call agarose gels, which is, again, is another form of gel electrophoresis that looks at different nucleic acids. So again, this is going to look at mostly DNA in our process in RNA as well. Similar to the SDS page, we have this ladder on the left-hand side, which is known number of base pairs of different fragments of DNA with known number of base pairs. And we can see that the higher up, the larger the fragment of DNA, the lower, the smaller. And what we show here is really a pretty interesting figure, where we have this time series of our process, and we can see that in the initial part of our process, we have a lot of nucleic acids in there. But as time goes and those different enzymes and detergents are helping to break down those nucleic acids, we can actually see that process unfold to where we have this literally less than 10 base pairs or less than 100 base pairs, sorry, fragments, which will easily filter out when we get to our filtration process. And so that's just shown here on the left 2 images. On the right-hand side, as I mentioned, we do PCR. One of the reasons we do PCR is to verify that any genetic modifications we make to our native microorganism is retained as we grow those microorganisms up. So if we insert a specific gene, we want to make sure that that gene is expressed as those cells grow. And so that's what we've done here is we go through the PCR process for PCR, which is amplifying. That polymerase chain reaction is going to amplify a targeted fragment of DNA. And then what we want to see is make sure that that band appears with our positive control. So we have a control group we know has that band, and then we verify that our samples have the same band for that particular DNA sequence or DNA fragment. And this again is going to help us confirm that whatever modification we made to the organism was retained. Last little technique that we go over is gram staining. This is again important in identifying any contaminants, whether in our upstream processing or downstream processing or in our final products. And so here on the left-hand side, this is where we have streaked a plate or we spread a plate. We can identify these different colonies, and then you see some of these colonies have unique characteristics where you have this large, very large colony. You have these smaller beige colonies, even smaller colonies, maybe a pink colony or an orange colony here and there. So what we do is we harvest those individual colonies, we streak them on a plate, and we perform gram staining. And this helps us to identify what those particular different organisms are based on the shape of the organism as well as how it's stained. So here we have a gram positive where the crystal violet has stained the membrane, and here we have a gram negative. And again, we can look at common contaminants to identify what those are and potential sources of those contaminants. So that's kind of the general overview of the microbiology lab and what we do. I think more relevant to you are what are the careers at Danimer and what education level, how can you coach your students or encourage your students in pursuing science degrees. So we do hire from high school level all the way up to PhD, but most of our laboratory positions are either an associate's degree or bachelor's degree, typically in a science field, biology, chemistry, engineering, biosystems engineering, something like that. I will say that we primarily hire bachelors, but there are associate's degree levels as well. So if you want to see any jobs that are available, here's a link to the Indeed postings as well as our LinkedIn postings. And then I guess going back to the careers at Danimer, you may have a lot of students that are saying, when am I ever going to use this? And hopefully you can use this as an example to show, hey, you can do this kind of science right here in your hometown and make a difference in the world by making this biodegradable plastic. We are, community engagement is very important to us, and this goes all the way up to our Chief Technology Officer, Phil Van Trump. He strongly encourages us to participate in community engagement, which ranges from internships for both students as well as teachers. Due to the current COVID pandemic, the internships have been limited. We're hoping that as things progress and the vaccinations and opening up, that we will be able to resume the internships potentially in the fall semester, but hopefully most definitely by next year. We do participate in school demonstrations, and I think I saw Kyle earlier on here. I'm not sure if you made it to this breakout room, but Kyle Kelly from Bainbridge High School invited us to come in and do some demonstrations. And as you can see here in the bottom left figures, we did a SDS page so that the students were able to do everything from pipetting to actually loading these gels and running these gels and seeing the protein content of our different proteases. And lastly, we do school tours, whether it was with a school or with a group. And again, this is kind of dependent upon the COVID-19 situation, but we're hoping that we can open our doors again here very soon and let you and your students in and see what kind of science we do here at Danimer. On the right-hand side, this is my contact information. I can't send you the full presentation, but if Megan or Kristen, I can send you this final page so that you'll have my contact information, my email address. Feel free to add me or follow me on whatever you do on LinkedIn. I'd be happy to communicate with you via LinkedIn. Danimer also has a Twitter, Facebook, and LinkedIn account. I encourage you to follow those as we continue to have news releases and seeing what other companies are interested in our products. With that, again, I want to thank you. Thank Megan and Kristen and the Georgia Bio Ed Institute for allowing me to present and meet with you. If we have time for questions, I'll be happy to answer any questions you may have.
Thank you so much, Matt.
I had a quick question, if I could ask.
Yeah, go ahead.
Yes, please.
Yeah, I'm just interested. The PHA, what's the breakdown time on that? How long does it take to biodegrade?
Obviously, there's several factors, humidity, moisture content of the area, temperature, and things like that. But typically, it's fully biodegraded in about 3 months. So this is something that potentially in your classroom, we could have a PHA product that you could put in some soil. I mean, most importantly, you got to have the bacteria there. So if you go outside and harvest some soil from outside, it should biodegrade within 3 to 6 months, but typically it's on the 3 month side.
Yeah, that's super cool.
Does your organization have like some lesson plans that you've already put together that you can share with teachers? Like you were suggesting, like taking some of those products already, they can put them in the soil in their classroom. But do you guys have something that you've already written up or worked with?
That's an excellent question. I'm not familiar with any of those. What I can do is I'll work with Phil and Carol here, and we can maybe come up with something like that, or at least be able to send some products that you could do that in your lab or in your classroom.
Great, thank you.
Awesome. Yeah, and Matt, we can help you guys with that if needed.
Yeah, perfect. Thank you, Megan.
Awesome. So a question in the chat, Emily Gunderson asked if there's a location in Athens. Is there one at UGA?
Yes, there is. So we do have a analytical lab up there that does our NMR spectroscopy. They also do the strain development up there. So all of any genetic modifications to the strain take place up there. Dr. Richard Eaton is the one.
Awesome. And then another question, what is the cost of PHA relative to single-use plastics?
Britt, that's an excellent question. I don't actually know the cost ratio there. What I do know is that obviously petroleum-based plastics have been around for a long time. They're the lowest cost, and what we're focusing on now is driving our costs down so we can be cost competitive. As I mentioned, one of the primary goals for us is to investigate new materials like enzymes and detergents, where we're trying to find the cheapest one that accomplishes our goal, but is also a green product. It's got to be sustainably made too, because we want to be a fully sustainable company. So I don't know the exact number right now. Petroleum-based is obviously beating us, but we are becoming more and more cost competitive, especially as we scale up and increase productivity.
Awesome. Cool. Well, thank you so much, Matt. I think we're out of questions for right now, but yeah, if you can share.
I was looking it up.
Oh, there's
straws.
Yes.
I'm sorry. You can get 500 of the straws from Walmart.com for about $10.
Okay. Yeah. And I guess something else to point out about the straws is, I don't know if you've ever used paper straws. I am not a fan. They tend to, get real soft and mushy kind of quick. So these are plastic straws. They're not going to biodegrade while you're using them. So this is a little more kind of normal type straw, but it also doesn't have the detrimental impacts.
So, I'm teaching AP Environmental, and my next unit in a couple of weeks is going to be waste management and, terrestrial pollution. You said you might be able to get samples to Megan and Kristen that could be shipped out to us possibly, and then I could put that right into the lab.
Yeah, I think that's something that's certainly feasible. I have to actually go through upper management to make sure that I'm not, breaking any of their rules, but I can't imagine that's going to be an issue, and we'll definitely try to get some of those to Megan and to you as well.
Okay, cool. Thank you.
If anything, you can run to Walmart and grab a box or something like that, but hopefully we can send you some products.
There you go. Awesome. Well, thank you, Matt. It is